Hi all, myself Naveed Ahmad. Today I am here to present a seminar on transplantation and graft rejection. I done this seminar under the guidance of Utpala ma'am. These are the contents of my topic. Let's move on to the introduction. Transplantation refers to the act of transferring cell, tissue or organ from one site to another site for the purpose of replacing or repairing the damaged or diseased tissue or organ. Many diseases can be cured by the implantation of a healthy organ or tissue from one individual to another individual by of transplantation. The cells, tissue or organ which are used in the transplantation are called graft. The individual who provide the graft, he become the donor. The individual who receive the graft, he become the receiver. The organ transplantation is a major operation which is offered only when all the treatment options have been failed. The immune system play a critical role in the transplantation. The complex mechanism of the immunity under normal circumstances work to identify foreign microbes and direct the immune system to destroy them. It poses a significant barrier to a successful transplantation. The rejection of the transplant occur in the instance where the immune system identifies the transplant as a foreign, triggering a response that will ultimately destroy the transplanted organ or a tissue. Types of transplantation. There are importantly four types of transplantation: autograft, isograft, allograft, and xenograft. Let's see one by one. Autograft. The tissue of the original donor is grafted back into the same donor. For example, the skin graft from the thigh to the face is severely deformed cases of burnt individual. The next one is isograft. The transplantation of an organ or a tissue between genetically identical twins is referred as isograft. Next one is allograft. The transplantation of an organ or a tissue from a donor to a non-genetically identical individual of the same species. The next one is xenograft. The transplantation of an organ or a tissue between two different species is referred as xenograft. Types of graft rejection. There are importantly three types of graft rejection. First one is hyperacute rejection, second one is acute rejection, third one is chronic rejection. Let's see one by one. Hyperacute rejection. Rapid from minute to hour. This type of rejection occur very rapidly, result in the necrosis of the transplanted tissue within minute or a few hours of contact. It always results from the reactivity of the donor cells with a pre-existing antibodies. Hyperacute rejection occur usually within the 24 hours of transplantation. It is characterized by the thrombotic occlusion and hemorrhage. The blood types and the xenograft are the best example for hyperacute rejection. Acute rejection. Days or week. Acute rejection usually begins after the first week of transplantation and most likely occur to some degree in all transplants. It is caused by the mismatches of HAL antigens, that is human leukocyte antigen. It is present in all the cells. The T cell mediated immune response directed against the foreign MHC molecule, the inflammation and leukocyte infiltration of the graft results. It's the most common type in the acute rejection. Chronic rejection is the third type of rejection and occur months to year after the transplantation. It is characterized by the graft atrial occlusion which result from the proliferation of smooth muscle cells and production of collagen by fibroblast. This process is termed as accelerated or graft arteriosclerosis, results in the fibrosis which can cause ischemia or a cell death. Mechanism of graft rejection. The process of graft rejection can be divided into two stages: sensitization phase and effector phase. Sensitization phase, in which antigen reactive lymphocytes of the recipient proliferate in response to alloantigen on the graft, but in the effector phase, in which the immune destruction of the graft take place. Here we can observe the process of graft rejection. In the first step, the antigen printing cells, for example, dendritic cells are the examples of the antigen printing cells which present the foreign antigen through an MHC molecule. When they interact with the T helper cells, they activate the T helper cells. The activated T helper cells goes on activating other cells through a factor called interleukin factors. There are a series of interleukin factors like interleukin 1, 2, 3, 4, 5 and 6. Here, in the first step, the TH cells activate TDTH cells, means delayed type of hypersensitivity TH cells. These activated cells produce interferon alpha and tumor necrotic factors, which produce lytic enzymes which can destroy the graft. In the second step, the TH cells activate TC cells, that is cytotoxic T cells. There are two types of the cytotoxic T cells are produced CD8 and CD4. This activated TC cells converted into cytotoxic T lymphocyte cells, which can damage the membrane of the graft. In the last step, we can observe that the TH activated TH cells converted into B cells. These B cells can produce plasma cells and macrophages or natural killer cells. These produced plasma cells produce many antibodies. These antibodies combines with a complement can cause the lysis of the cells, which result in the destruction of the graft take place. By this process, the graft rejection occur. Antigen involved in the graft rejection. The ABO blood group typing antigens and MHC molecules are the important antigen that are involved in the graft rejection. The mismatches of the antigen and antibody result in the lysis of the cells take place, and the mismatched MHC molecule between the individuals result in the graft rejection take place. Allorecognition. There are two methods of allorecognition: direct method of allorecognition and indirect method of allorecognition. In the direct method of allorecognition, it is rapid from minute to hour. Here, dendritic cells act as the antigen printing cell. They present the antigen into the B cells through MHC molecules. Here, T cell recognizes unprocessed allogenic MHC molecule on the graft antigen printing cells. Indirect allorecognition. It is humoral or cell mediated. It takes days to week. Here, dendritic cells act as antigen printing cells. They taken up the foreign antigen and process it and present it to the T cells. So here, T cell recognizes the processed peptide of allogenic MHC molecule bound to the self MHC molecule on the host antigen printing cells. Prevention of the graft rejection. There are four important methods for the prevention of graft rejection, namely familial grafting, cross matching, antibody mediated microcytotoxicity test, and mixed lymphocyte reaction. Let's see one by one. Familial grafting. Major histocompatibility complex or HAL in human is the major barrier for transplant acceptance. But due to the inheritance patterns of HAL genes, the transplantation within the familial members reduces the allelic mismatches significantly. Generally, there is a very little crossing over within the locus, and whole locus is inherited. Thus, there is a 50% matches of HAL alleles if the transplant is from parents to sibling, whereas it is 25% match from sibling to sibling. Hence, familial grafting ensures better graft acceptance. Cross matching. Cross matching is done before transplantation to check there is no preformed antibodies in recipient serum for donor HAL. In this method, serum from the potential recipient is mixed with a lymphocyte of a donor in the presence of complement for evaluating lysis or stained with a fluorescent to human immunoglobulin and assessed by a flow cytometry or fluorescent microscopy. The positive fluorescence or dead cell indicate the presence of anti-donor antibodies, which may lead to the hyperacute rejection of the graft. Antibody-mediated microcytotoxicity test. The presence or absence of the various MHC alleles can be detected by using this test. In this test, WBC from the recipient and donor are taken in a various wells on the microliter plate, and antibodies against various MHC alleles are added along with the complement protein. The lysis or cytotoxicity is assessed by using vital dye such as trypan blue. If WBC expresses a particular MHC molecule for which a specific antibody is added along the complement, the cell lysis taken place, which is further detected by the uptake of trypan blue. Hence, HAL typing can be done by using this test in order to detect histocompatibility. Mixed lymphocyte reaction. This test is used to quantify the degree of compatibility in MHC class 1 of donor and recipient. The lymphocytes from the recipient serve as the responder cells and the donor cells act as a stimulator cells. The stimulated cells are X-ray irradiated and treated with a mitomycin C. The T cells activation is indicated by the proliferation of the recipient T cells. It is measured by the intake of H3 thymidine. The intense proliferation of the recipient lymphocyte indicates the poor prognosis of survival of the graft. Finally, I sum up my topic. Transplantation refers to the transfer of cell, organ, or tissue from one site to another site for the purpose of replacing or repairing the damaged or diseased tissue. Many diseases can be cured by the implantation of a healthy organ or tissue. Transplantation is a major operation which is offered only when all the treatment options have been failed. There are four types of transplantation, namely autograft, isograft, allograft, and xenograft. Only autograft and isograft are accepted. Allograft and xenograft are generally rejected. Blood group antigens and MHC molecule involved in the graft rejection. The recognition of the transplant organ by direct and indirect method. They detect and reject the transplanted organ. Rejection of the transplant occur in the instance where the immune system identifies the transplant as a foreign, triggering a response that will ultimately destroy the transplanted organ or the tissues. These are my references.